Okay. Hi everyone, my name is Emily. I'm a PhD student in the biomedical engineering program at the University of Calgary, and today I'm happy to share my work on the controlled and rapid expansion of equine bone marrow-derived mesenchymal stromal cells in stirred suspension bioreactors, which is part of a transdisciplinary approach to develop stem cell therapies for equine cartilage injuries. So mesenchymal stromal cells, or MSCs, are commonly used throughout veterinary medicine due to their trilineage differentiation capabilities, their potential for tissue regeneration, as well as their immune modulatory effects. Specifically, bone marrow-derived MSCs offer a promising autologous cell therapy, as they're relatively easy to isolate from the large volumes of marrow available from equine patients. However, although there's relatively positive outcomes of using these MSCs in joint injury models, there lacks a consensus on the techniques used in these cell therapies, such as cell line derivation, cell expansion, and transplantation. So in this study, we aim to use equine bone marrow-derived MSCs to treat complex joint injury in equine patients. This is done using three aims, the first being the generation of a reproducible model of equine anterior articular soft tissue injury of the stifle by my co-author Kasara, who has a poster on this later today. The second being the design and optimization of a bioprocess for expanding MSCs in stirred suspension bioreactors, and the third being a preclinical trial, which is currently underway, to combine these first two aims by re-injecting autologous bone marrow MSCs into this injury model and quantifying the effects using gait analysis, histology, and biomechanics. However, today I'll only be presenting on the second aim of this study. So when designing a bioprocess, there are many different things to consider. The first being your inputs, such as your cell source, the media required, any supplements, as well as microcarriers if the cells depend on it. You also need to consider your method of expansion, as well as any purification steps for cell use. So traditional methods of expanding these cells use static culture, which, however, prevents efficient scale-up. Rather, we must scale out into multiple vessels, which increases heterogeneity, labor, and time required to produce clinically relevant cell densities. Dynamic culture, specifically bioreactors, offer a scalable platform to expand cells, and they also offer inline process monitoring and computer controlled in larger vessels. So in this specific study, we knew our cell type, which are autologous equine bone marrow-derived MSCs, our media, which contains 10% serum and 2 nanograms per mil of FGF, and our culture vessel, which was a horizontal blade stirred suspension bioreactor at the 0.1 to 0.5 liter scale. So as MSCs are an adherent cell type, they require a surface to attach to and grow on. And in dynamic culture, these are microcarriers, which are small beads which can be customized with different surface coatings, matrix materials, porosities, surface charge, and sizes to induce the attachment of specific cell types. So here I've included an image of how cells typically grow on traditional static culture, as well as fluorescent nuclei of cells grown on microcarriers. So in order to determine the optimal microcarrier for this specific cell type, a high-throughput, small-scale screening was conducted in non-adherent well plates on an orbital shaker to simulate dynamic culture. A total of 8 commercial microcarriers were screened with varying surface coating sizes and charges, and the results were quantified using confocal microscopy and an Alamar Blue cell viability assay on days 1, 3, and 5. Here we can see the day 5 results, and what we were able to do was identify 2 potential microcarrier candidates for dynamic culture, the first being collagen-coated microcarriers, and as an animal component-free, we also looked into plastic microcarriers. So to determine the best microcarrier for true dynamic culture, I scaled these microcarriers up in 100 milliliter stirred suspension bioreactors, and what we were able to conclude was that collagen-coated microcarriers allowed for the greatest amount of cell proliferation in dynamic culture. However, a cellular fold increase of only 2.5 needed to be optimized in order to reach the clinically necessary cell densities. So in order to do this optimization, I performed what's called a factorial design of experiments, or DOE, where we were able to optimize specific bioprocess factors to have the highest cellular fold increase. So the bioprocess factors chosen were the surface area to volume ratio, or the microcarrier density, the starting cell density, as well as the cell feeding regime. And each of these were tested at a high and a low level, resulting in 8 total conditions. And then our output was cellular fold increase in order to standardize for the varying number of initial cells in each of these reactor conditions. So each of the 8 conditions were run in parallel over a period of 9 days, and the day 7 cellular fold increase was taken as the output variable, as it was before any of our conditions began to dramatically decline. Here I've included the confocal images and cellular fold increases on day 7, and an ANOVA analysis revealed that one of our conditions was statistically higher than all others, which was a low surface area to volume ratio, a low starting cell density, and a 50% media change on day 3. Further statistical analysis was performed using JUMP, which looks at the significance each variable has on our output variable by making cellular fold increase a function of these 3 bioprocess variables. So what we can see here is that increasing the cell seeding density and microcarrier density have an inverse correlation on our cellular fold increase, whereas adding a media change has a positive correlation. However, this media change has the lowest significance on our final fold increase, with the initial cell seeding density having the highest significance. From here, we were able to design our optimized bioprocess for this preclinical trial. We extract the bone marrow on day 0 and then derive a cell line from this marrow over 2 weeks. And after the cells undergo their first scrape passage, approximately 1 million cells are seeded into a 100 milliliter stirred suspension reactor, cultured for around 5 to 6 days, passaged into 2 250 milliliter reactors, cultured for another 5 to 6 days before the cells are purified and ready for use. So in order to validate this process before moving on to a preclinical trial, we ran it with 3 pilot donors, 2 mares and 1 gelding, all of various ages, and we actually saw quite a bit of variability between the 3 donors. However, with this optimized process, we're able to reach the 40 million cells required for reinjection within 12 days. So far, we've performed 3 of these preclinical trial runs on 3 donors. We're hoping for a total of 9. So here I've included the imaging as well as daily bioreactor counts over the 2 bioreactor passages, as well as the immunophenotype, which we validate after each passage to ensure the cells are not changing at any point of the bioprocess. And before re-injecting the cells, we do a mycoplasma test, with all 3 donors having come back negative. To ensure the cells maintain their functional abilities, we did trilineage differentiation after this bioprocess, with the cells still being able to maintain their ability to differentiate into cartilage, bone, and fat. So by developing this bioprocess in parallel with this experimental injury model, we were able to ensure that we could produce a clinically relevant cell density within a clinically relevant timeline. So if you're curious, this is what 40 million cells looks like. And then here was one of the coolest moments of my life as a grad student, which was watching Kasara inject the cells grown in a bioreactor into the medial femoral tibial joint of one of our patients. Future directions of this study, we hope to eliminate the use of serum in order to decrease batch-to-batch variability and reduce risk of immune rejection. There's also the Center for Cell Therapy Translation, or C2T2, which is currently under construction at the veterinary school at U of C, which will allow for more trials like this, combining biomedical engineering, stem cell research, and veterinary medicine. And finally, I've shown quite a bit of variability between donors today, and a possible way to overcome this in the future would be the large-scale manufacturing of a universal donor, which my colleague will touch on in the next presentation. Finally, I'd like to acknowledge all of my amazing co-authors for all of their guidance and hard work in this project. I'd also like to thank my lab members in the Kalos and Krawitz labs, as well as the Sparks lab members, Mike, Tilo, Brent, and all of our research horses, as well as my funding sources, and I can now take any questions.
Thank you so much. That was a really nice. Any questions?
Hi, thank you for sharing this amazing work. I have more technical questions and comparison questions. So, how do you scrape off your stem cells from those microcarriers?
So to remove cells from microcarriers, we do a series of washing steps, and then we add an enzyme we use like recombinant trypsin or triple E to remove cells from the microcarriers while they're spinning, and then we just separate the cell suspension from the microcarriers for use.
And in terms of comparison to monolayer expansion, which would be pretty straightforward, kind of multiple lab already, mastering this, comparing to yours, maybe more laborious, more training would be needed, or it's easy?
Well, I mean, this is my life, so to me it seems straightforward, but when you think about it, I think that static culture can be more laborious when you think of how many passages are required and how many different vessels there are to keep track of. With a reactor, there's a day of work setting it up and a day of work kind of when you're purifying your population, but you have one vessel well mixed that you're able to monitor throughout the growth process, which in my opinion kind of makes this process less laborious.
Okay, last question. You can go.
Very nice job.
Oh, thank you.
I worked on this similar project a number of years ago and found that when I did all of my comparisons with spinner bioreactor versus monolayer, and in terms of the expansion rate, in my hands, the monolayer was way faster. Did you look at the difference between doing spinner against monolayer as well as between two different microcarriers?
Yeah, I always have a static control going as well, just to make sure that, if we see a difference in immunophenotype or cell growth kinetics, if that's the same in static or if it's a bioreactor problem. But with the optimization, I found that we've had a much higher fold increase after that optimization. It's just understanding how to kind of make those factors work for you to produce the highest cell density without changing any of their functional characteristics.
Thank you so much.
Thank you.